Hello, everyone, and thank you so much for being here. I'm really excited to present the research I've been doing for the past few months, especially since this is my first summer doing research as a college student. So before I start talking about my project, I want to give a brief overview on meiotic recombination. Recombination is an integral part of meiosis, as well as chromosomal separation within eukaryotes. The process begins with a series of double-stranded breaks, which form, or DSPs, which form in scattered hotspots throughout the chromosomes. These DSPs get repaired into non-crossovers and crossovers. And crossovers are really important since as meiosis progresses, as you can see up here, the chromosomes actually physically overlap and exchange genetic material at these crossover sites, such that the resulting offspring is genetically distinct from either parent. And this entire process is very important for genetic diversity. However, currently, there is no way to directly identify which specific double-stranded break will result in a crossover. And crossovers actually form much more selectively than double-stranded breaks. You can see double-stranded breaks form just scattered everywhere throughout the chromosomes. However, crossovers are limited to very specific parts of the chromosomes. And in maize in particular, a single cycle of meiosis can lead to up to 500 double-stranded breaks. However, only around 10 to 20 of these actually progress into crossovers. And this is mostly due to the fact that crossovers typically have a very different epigenetic environment to typical double-strand breaks. And if we could predict which crossover sites if we could predict which double-stranded breaks turn into crossovers, this would be very revolutionary for plant breeding as a whole. But yes, as I mentioned before, DSPs and crossovers have very different typical environments. Typically, double-stranded breaks are characterized by forming in highly methylated intergenic regions, so they're relatively spaced apart from genes. Whereas crossovers are typically characterized by low methylated genic regions, so they're a lot closer to genes. However, DSPs that turn into crossovers are in fact located in an environment similar to that of crossovers. So the main goals of our project was to computationally predict the fate of double-stranded breaks using various epigenetic and genomic features. We hypothesized that using this assortment of features, we'd be able to partition a data set of DSPs into two main clusters, which would correspond with features associated with DSPs and crossovers respectively. Namely, the features that we looked at in particular included epigenetic features such as DNA methylation, histone marks, as well as nucleosome occupancy. And we also looked at some genomic features as well, including GC content, distance to the closest gene, as well as replication timing. And all of these data were obtained via the Pawlowski lab's prior experiments on maize, as well as supplemental information from Maize GDB, which is an online database filled with information on maize. So in addition to all of those features, we also had various data sets to use. We had, of course, our treatment group, which was the double-stranded breaks. However, we also had a positive control in the form of identified crossover regions, as well as a negative control in the form of just random genomic intervals. For our experiment, we mainly used unsupervised learning. This framework, unlike supervised learning, does not require labeled data, so we don't need to know which DSPs turn into crossovers, which, as I mentioned before, we do not know. Instead, unsupervised learning analyzes hidden patterns within the data set as a whole and pieces together the relations between features in the points. Here's like a very simplified graph, or very simplified figure showing the process of unsupervised learning. You can see here, each of these dots is not labeled. However, they still have very different features, which the analog is colors. But then it goes into the algorithm, and the algorithm is able to process it by analyzing these features and the differences between them into these output clusters. And clustering is actually just one type of unsupervised learning. There's also dimensionality reduction, which we used alongside it, in the form of UMAP, which stands for Uniform Manifold Approximation and Projection. And basically, UMAP functions by taking in a data set with multiple dimensions. In our case, our initial double-stranded break data set had like 12 dimensions because of all of the features. However, it specializes in condensing this massive 12-dimensional data set into just 2 dimensions, which can easily be plotted on a 2D plane. And it also clusters groups based on having similar features. And actually UMAP's very special in that not only is it really fast and really effective, but it also preserves distance across dimensions, which made it a prime candidate for our study. In addition, we also use density-based clustering, which basically it's able to identify distinct clusters based on the number. So yeah, now I'll talk about our main results. So we ran UMAP and the density-based clustering on the full DSP dataset with all of the features. And we found that the 2 algorithms split it apart into 5 main clusters, as you can see here. And you can also see one of our first observations was that one of the clusters was much larger than all of the others, and it actually had 90% of all of the DSPs. But we wanted to look deeper, and we wanted to analyze specifically how the clusters differed in terms of features, and if there were any that stood out to us. So looking into cluster 1, we plotted the average feature per each cluster, and basically using this, we were able to compare it to the averages for the DSPs and see relations. And as you can see, cluster 1 is very much analogous to the expected environment for double-stranded breaks. It has very, very similar features all across the board. And actually, clusters 2, 4, and 5 also had very similar feature layouts with a lot of similarities to the double-stranded break environment. However, some of them had like 1 or 2 differences, but overall they were very similar. Cluster 3, on the other hand, has a very different genetic layout. And we can see here that in particular, the methylation data and the distance to the gene data were much less than in the previous slide. And there were also some minor differences across the board, most of which were very similar to the expected crossover environment. And this was really interesting, considering that cluster 3 was actually comprised of almost 2,000 points. So these data, it's not just a few outliers. It is actually like cluster 3 is like over here, you could see. So yeah, we don't know if a specific we don't have a way to find if a specific DSP turns into a crossover or not, but we were able to see that cluster 3 had very similar features to actual crossovers. So we wanted to see if the actual crossovers themselves would fall into cluster 3, because as I mentioned before, crossovers and crossover competent points have very similar features. So we ran the UMAP and clustering algorithms on a combined data set of the DSPs and the crossovers, and found that the vast majority of the crossovers, 97% of them in fact, were categorized as cluster 3. And you can see they're marked in dark blue here. And this was really interesting. We also just tested on the negative control, which, as I mentioned before, was comprised of just random intervals, just to see if we'd see a similar pattern there. We didn't expect one to happen, and as you can see, there wasn't really one. For like the random intervals, just the majority of them just formed into this one little pseudo cluster in the middle, which isn't seen in any of the other clusters. So, but yeah, you can see there's not really a relation between the random intervals and the double-stranded breaks, which makes sense. So in conclusion, the features that we used were indeed able to produce clusters. And one of those clusters had a very similar genomic feature layout to crossovers themselves, and also represents a very similar fraction of the total DSPs. Um, DS crossovers are generally around 2 to 10% of total DSPs, depending on how many DSPs there are. And these data support this overall statistic, since, as you saw before, it was the cluster 3 was actually around 5% of the total DSPs. And these data suggest that crossover 3 represents, or at least is closely related to, crossover competent DSPs. And for looking into broader applications of this information could be used to artificially induce crossover events potentially in plants if we know more about like the features associated with differentiating DSPs. And potentially this could help plant breeding in the future. However, that's well beyond the scope of this project. As for further steps for now, there's one application in particular that we actually really wanted to do, but unfortunately we were a bit short on time come the end. And that was that previously, in the beginning, we weren't able to use supervised learning or supervised machine learning, since we didn't know which DSPs turn to crossovers. However, now we do know which DSPs turn into cluster 3, which the data suggests is strongly related with crossovers. So now that we know which DSPs are likely to result in crossovers, we could potentially utilize supervised machine learning, or maybe even, we were actually thinking of using random forest, but again, we were running short on time, in order to refine our findings or even look more in depth. However, that is mostly all for this project, and I would like to thank a lot of lovely people for all of their help and support throughout this project. This could not have been possible without any of them. So yeah, thank you so much. Now I'll open it for questions. Yes?
So what do you think? What are the most important features you use in your analysis that separates your cluster 3 from the other clusters?
To answer your question, we found that the methylation data, as well as the transcription start and end distance to the transcription start and end, were most prominently different. Like, so clusters 1, 2, 4, and 5 all had pretty much this layout with some minor changes. But one thing that remained constant within all of them except for cluster 3 was CG and CHG methylation. And that definitely lines up with our current understanding on DSPs forming in highly methylated areas and crossovers forming in low methylated areas. Yes?
I would just like to know why you use UMAP in comparison to PCA, like the benefits of cost.
UMAP is actually fairly recent in development, and however, it is especially useful in terms of speed and accuracy. And like one of its main defining characteristics is the fact that it preserves multidimensional like distance when reducing the dimensions. And yeah, that's one of the main reasons. Yeah.
So if I wanted to in artificial crossover side of the base genome, would you be able to tell me what I need to do at that site to make crossover?
Of course, there are a lot of once again, this is a very broad application. This isn't something that could feasibly happen as a next step, like a direct next step for this project, but it would need some intermediate steps first. However, like knowing which features are associated with crossovers, such as being able to lower methylation levels or being able to potentially like I think in this regard, you'd have to look at epigenetic features more so than like genomic features, since you can't really, because distance to a gene is one of the features, though again, like it's not it's not like um it's not like even in cluster 3 and the crossovers, you could see that there's a wide variety of different, like you could see the standard deviation is considerably big for transcription start and end. So, but like that's like for transcription start and end, you can't really like move a gene somewhere else or like, however, for things like methylation or something, presumably you'd be able to artificially, well, in the future when at least, you'd be able to like artificially like methylate a certain point near a double strand break. If that answers your question. Yes?
A possible explanation for the huge standard deviation plus the
Oh. I believe that the reason for that, especially for transcription start and end, was because there I don't it's because the transcription start and end data were from like all across the like there wasn't really like, although a lot of the although like especially compared to like cluster 1 and DSPs as a whole, you can see that there's a lot more there's a lot more typical distance to the transcription start. However, even in crossovers and cluster 3 by proxy, there's still some amount of variation for how far a crossover can be from transcription start. And then, if that answers your question.